Hello, everyone. Welcome to week 2 of our course, and this is the second part of chapter 1. So as I mentioned in last week's video, I'm going to keep these videos to about 15 minutes, only longer if necessary to cover a topic that's maybe a little harder or maybe not covered that well in your textbook. So this will be about 15 minutes. The second part of this chapter deals with naming and classifying organisms. So what does that mean? We think about all of the life on the planet. There's they say, about 1.7 or 1.8 million species that have actually been identified as unique. That does not mean that we've named all of the species on the planet. Some people estimate that there's about 10 million species on the planet. So we've only begun to even scratch the surface of the diversity of life. So imagine you have 1.8 million objects. How are you going to organize that? So what we do in science, it really was started by Carl Linnaeus in the 1700s, is that you start looking at the characteristics of the organisms and you begin grouping them with things that they are like. Makes a lot of sense, right? We do things like that on your computers with your file folders or so on, or the apps on your home screen. You've organized them into different types of apps. Either it's for your pets or it's for shopping, it's for banking. So we call this taxonomy. It's the science of classifying or essentially organizing organisms. And each of the groups that we're going to talk about, from the domain all the way down to the species, is called a taxon. Okay, so let's talk about nomenclature, classification, and identification. All right, so nomenclature, that's a fancy word for naming things. So scientific names and categories. Essentially, we know, just like you all have your formal name, your first name and your last name, scientific names also have a two-parted name called a binomial with a first name and a last name. The first name is a genus. It can be shared among many types of organisms. So, for example, I'm a botanist by training, and I like oaks. The genus of an oak is Quercus. So there's red oak, Quercus rubra. There's white oak, Quercus alba, and it goes on and on and on. So the first name is the genus. It's a little bit like our last name. So in our big expansive family, we've got Powers, we've got Dawn, we've got Powers, we've got Heidi, we've got Powers, we have Maggie. Okay, so that Powers, that last name, is like a genus. It's one kind of smaller grouping, but it's identifying a small subset of life. Okay, the second part of the name is the specific type of the genus. So rubra for red. Think about it. Red doesn't tell me much, but it certainly tells me which species of the genus Quercus. Alba means white. It tells me the specific species of a genus. Okay, so back to some family names. Powers being the genus. And their first names are just telling you which one, whether it's Maggie or whether it's Michael or whether it's Colin or whatever. So these formal names in science are called the scientific names. Okay, a two-parted name. We also can say that that's the species name. These names are often Latinized, and they're often comprised of words that help us characterize the particular kind, like red in the case of Quercus rubra. Let's take a look at an example of a bacterium. It is a beneficial bacterium in your gut called Escherichia coli. You probably know it best by its truncated genus name, just E. coli. The genus is Escherichia, and there are many different species within that genus, right? Just like a last name, many different individuals in a family. The specific epithet is coli, so it's telling us which one of the Escherichia genus. So when we take a look at the word, you're going to see that I've italicized it. I have capitalized that first name, the genus, and I have kept the first letter of the specific type, the specific epithet, lowercase. This is how we will type scientific names. So when you have to turn any lab reports in or things like that, and you need to include the scientific name or the species name, make sure that you write it correctly or you type it correctly. Typing it is always in italics. Now, we know that sometimes we turn in papers or labs and things like that, we might turn them in hard copy. And when we do that, we need to write the name in cursive and then underline it. If you don't write in cursive, then you would just simply write it in script and then underline it. You notice that the example Escherichia coli has now been written with its underline. All right, moving on. So nomenclature, the naming of organisms on the planet, really has come a long way since the advancements in DNA technology. So 1980s and really 1994 in particular, there was a lot of advancements in what we call DNA technology. So we were able to not only take a small amount of DNA, replicate that DNA by what we call PCR, polymerase chain reaction, and amplify the DNA. So essentially making lots and lots of copies. When you make a lot of DNA, now we can begin to really research it. So it's once that we developed the techniques in order to better study molecular biology that we were better able to name and organize life on the planet. So before, we really relied on external features and maybe reproductive characteristics and cellular features if we had the advancement of the microscope. So essentially, now we see that the organization of life on the planet is built on phylogenetic relationships based on their genetic information, their genetic heritage. And really, this is a byproduct of evolution. So now when we see how life is organized, we really begin to understand when they appeared in the fossil record, what kind of changes happened to separate them from other taxa. Okay, moving on. You can see the names are often descriptive. So when you see Staphylococcus aureus, a very common skin bacterium, that staphylo, or it comes from the Latin word staphule, means bunch of grapes. So anytime we have staph, it usually is relating to clusters of cells. Lacto for lactobacillus is telling us lacto means milk. Bacillus is the shape of the cell. So it's given us some indication of the features of the organism right in its scientific name. All right, classification. This is how we're organizing life from very broad groups, general all-inclusive taxonomic groups, down to the smallest, most specific. The smallest, most specific that we have talked about thus far is the species. So let's move on to what we now understand best as how we should organize living things. This came into being, I believe, in the late 1980s. Not me, but individuals, Woese and Fox, came up with the understanding that there were actually 3 origins of life on the planet, and we put them into domains. So we have bacteria, which is the kind of true bacteria. When you think about bacteria, you're going to think about the organisms in this domain. The archaebacteria are those things that are the ancient bacteria. They grow in the thermal vents. They grow in bogs where there's a high methane environment. They grow in things like hot springs and geysers in Yellowstone National Park. The last domain is made up of those cells we know as the eukaryotes, and hence the name is eukarya. So we have all the other life on the planet falls into that category. Let's move on. Okay, so if we're going to go ahead and talk about the broadest, most inclusive taxonomic group, this is the domain. And with the domain, we narrow it down a little bit into the kingdoms. We're going to talk about that on the next slide. Within the kingdom, there is a phylum. In plants, sometimes they will refer to that as a division. Within a phylum, there is a class. Within the class, there's an order. Within the order, a family. Within the family, a genus. And as we discussed previously, the genus is going to have several different species. So when we talk about the six kingdoms of life, we notice that the archaea and the bacteria, being in their own kingdoms, are also in their own domains. The other kingdoms of life are eukaryotes. So then we have things like plants, protists, animals, and fungi. And there we go. We have organized life into 3 domains, And in each of those domains, particularly the domain eukarya, we have our 4 kingdoms. If you're interested in the classification of, here we have humans on the left-hand side and classification of Paramecium, a protist on the right-hand side, you can see all the way down the kingdom, phylum, class, order, family, and then genus, species at the bottom. So it kind of gives you an idea. For example, the kingdom Animalia has all kinds of things from sponges to gorillas and starfish and humans. And each time we go down to a lower level in taxonomy, then we're going to have fewer different kinds of life because we've separated them into different groups, okay? Until we get to the bottom, the most specific one being the species. So I do want to urge you to look at the video link in your lecture guide. It talks about the Human Microbiome Project. So to go back to the late 1980s and 90s when we developed DNA technology tools to study molecular biology. After we were fastidiously working on the Human Genome Project, we began working on the Human Microbiome Project. So it used to be thought that, all bacteria were bad, but now we know that we are likely made up of more bacteria, virus, and even other like eukaryotic organisms in our body than our own cells, okay? So essentially, they swabbed different parts of the body from different test subjects and began to understand the community, if you will, of the microorganisms that lived in different parts of the bodies. And it turns out that they're slightly different. We'll come back to that. Here's some terms that you're going to hear and you need to know. So your normal microbiota. So you're healthy, you're an adult. You generally don't need antibiotics. Maybe once every 8 or 9 years you might have an infection, but essentially these are your own microorganisms that are living in your body and protecting you from outsiders. We would also call some of these resident microbiota. So these are the ones that are typically there and they're living and growing. They're tightly attached, for example, on your skin. These are resident microbiota. It's not really easy to get rid of all of them. Now, transient microbiota, these are the things that we can pick up temporarily. So we, grab a knob to a door, and you essentially then pick those up on your hand. When you go inside, you're about to eat dinner, you wash your hands, you wash them off. Okay, so these are loosely attached and easy to get rid of. So the microbiome research, the Human Microbiome Project allowed us to really understand that some normal microbiota are potential pathogens, but generally speaking, most of them are pretty good for us. And they colonize areas of our body and prevent other infections from taking hold. You're also going to see that there's some intestinal microbiota that can influence health. So you might have heard that there is a gut-brain connection. We will get into that more in the future, but essentially this is like the cutting edge research in microbiology. How is our microbiome or dysfunction of our microbiome causing diseases? Did want to briefly tell you about how do we become colonized with bacteria to begin with. So when a baby is developing in utero, essentially they're getting small amounts of microorganisms, but not a lot. And then when those babies that are born through the birth canal, they pick up a tremendous amount of bacteria, mostly the genus Lactobacillus, from the birth canal of the mother. Then as the child is getting older, they're picking up microorganisms from breastfeeding or from crawling and sticking things in their mouth. And then in late infancy, they have a pretty complex microbiome. So there's always the question, what happens with those babies that are born by C-section? Well, they will essentially catch up, but it takes about 3 years. Okay, so let's take a quick look. When we take a look at different bacteria in the body, okay, so you may have needed to pause that last slide so that you could look to see what areas of our bodies have high numbers, what areas have moderate and low numbers. So when we take a look at various parts of the body, we get an idea, if you take a look at the pie graphs for swabs that were done in different areas of the body, that essentially the microbiome is different. So different colors in the pie graph. So this is one of the results of studying the human microbiome. I thought I had one more slide. Shoot.